You pick up a book. Pages turn, words flow. You think nothing's changed. But inside your skull right now, networks are firing that weren't active 5 minutes ago. Physical cables in your brain are reorganizing themselves. And if you keep reading tomorrow and the day after, those changes become permanent. MRI scanners caught this. Scientists mapped it. The data is shocking. Frequent readers don't just know more words. Their brains are structurally different. White matter bundles thicker, gray matter regions larger. Neural highways that connect language areas to memory centers, to visual processing zones, stronger and faster. This isn't motivational talk. This is documented neuroscience from 25 years of brain imaging studies. The scanners don't lie. Tell you something. If you value documented neuroscience and real brain imaging findings like this, subscribe. It's the best way to support our work. Also comment from which city you're hearing this from. This script is researched and factual using sources from peer-reviewed journals in Nature Communications. Biology, published November 2025, showing white matter functional connectivity changes. And from the journal Developmental, Cognitive Neuroscience, February 2024, issue documenting structural neural connectivity in pre-reading children tracked longitudinally with diffusion MRI. These aren't popular science magazines. These are primary research papers where scientists put their careers on the line with every claim. Now back to the information. That's when the first structural MRI studies linking reading habits to white matter began appearing in neuroscience journals. Researchers at universities across the United States and Europe started asking a simple question. Does reading change the brain, or does the brain determine reading ability? They needed longitudinal data. They needed to scan the same people multiple times over months or years. They needed control groups. They got all three. Between 1998 and 2024, 89 peer-reviewed neuroimaging studies examined white matter changes related to reading behavior in children and adolescents aged 0 to 18. Published in May 2025 in the journal Children, this systematic review pulled every relevant study. The finding? Children with stronger reading abilities consistently showed increased fractional anisotropy in key white matter pathways. Fractional anisotropy measures how organized the white matter is. Higher numbers mean the neural cables are tighter, faster, more efficient. The arcuate fasciculus, that's the white matter bundle, connecting language production areas in the frontal lobe to language comprehension areas in the temporal lobe. Readers had thicker arcuate fasciculi, the superior longitudinal fasciculus. That's the highway connecting frontal cortex to parietal cortex, supporting phonological processing and reading fluency. Readers had denser, superior longitudinal fasciculi. These aren't small differences. These are measurable structural changes visible on a brain scan. He published the first studies in the world using MRI to show relationships between reading habits and brain structure in preschool-aged children. Not teenagers, not adults. Kids aged 3 to 5. Why that age? 80% of brain development happens before age 3. But between 3 and 5, the brain is still massively plastic, still wiring itself based on what the child experiences. Hutton used diffusion tensor imaging. That's a special type of MRI that looks specifically at white matter. White matter is made up of fibers distributed into bundles called tracts. These tracts form connections between brain cells and the rest of the nervous system. Lack of development in these cables slows the brain's processing speed. But studies show reading, learning a musical instrument, or juggling improves the organization and structure of white matter. Hutton's team recruited 47 brain-healthy children, 27 girls, 20 boys. None had started kindergarten yet. Before the MRI, children took cognitive tests, 4 assessments measuring vocabulary, language processing, and early literacy. Parents filled out questionnaires. How much do you read to your child? What's the diversity of content? What's the quality of interaction during reading sessions? Do you just read, or do you ask questions and discuss? Results came fast. Children whose parents reported higher reading frequency, greater diversity of books, and more interactive reading sessions showed more organized white matter throughout the brain. The images were stark. One scan showed fractional anisotropy in blue for a child, with higher screen time and lower reading exposure. The white matter looked scattered, multidirectional, disorganized. Another scan from a child with daily reading showed tighter, more directional white matter bundles. Water molecules in white matter move along the fiber tracks. In organized white matter, they move in one clear direction. In disorganized white matter, they scatter. The MRI measures this. It's called diffusional anisotropy. Published November 2019 by CNN Health covering Hutton's research. The study was small, 47 kids, but the effect size was large enough to detect significant differences. 41 children tracked from the end of kindergarten through middle of grade 5. 3 scan time points. End of kindergarten, end of grade 2, middle of grade 5. They used a method called percent brain volume change with scaling adjustment. This accounts for the fact that children's heads are still growing. You can't just measure volume change because the whole skull is expanding. You need to separate reading-related cortical development from overall growth. The method works by calculating local morphological differences between brain scans at 2 time points while considering global head growth. What they found: during the first 2 years of primary school, gray matter regions of the reading network matured significantly. Left temporal regions, left temporoparietal regions. These areas are responsible for phonological processing and sound to letter mapping. Volume changes weren't random. Children who became better readers showed specific patterns of cortical thinning in these regions. Wait, thinning? That sounds bad. It's not. During brain development, cortical thinning in specific regions often reflects pruning of unnecessary connections and strengthening of important pathways. It's a sign of specialization and efficiency. Published in the same study, children with dyslexia showed deviant cortical development, but not in the reading network. Their differences appeared in regions outside the typical reading areas, suggesting compensatory mechanisms or different developmental trajectories. Instead of tracking children, they wanted to see if reading changes adult brains. 19 college students, all native English speakers, all skilled readers. The experiment ran for 19 consecutive days in July and August 2011. 5 days wash-in period where students came in for resting state functional MRI scans but did nothing else. Then 9 days where students read 1/9 of a novel each evening and returned the next morning for an MRI scan. Then 5 days washout where students stopped reading and continued getting scanned. The novel was Pompeii by Robert Harris, historical fiction based on true events, classic narrative arc. Every morning after reading, students took a quiz about the previous night's section and rated how aroused or engaged they felt. The MRI wasn't scanning them while they read. It was scanning their brain at rest the morning after reading. Resting state functional MRI measures spontaneous fluctuations in blood oxygen level in the brain when you're not doing any task. It reveals intrinsic functional connectivity between brain regions. On days after reading, significant increases in connectivity appeared, centered on hubs in the left angular gyrus and supramarginal gyrus. These regions are associated with perspective taking and story comprehension. Also increases in right posterior temporal gyrus. The changes exhibited a time course. They peaked the morning after reading. They began to decay after students stopped reading, but they didn't disappear immediately. Some connectivity changes persisted for several days after the novel ended. Published in the journal Brain Connectivity, December 2013. Lead author Gregory Burns, neuroscientist at Emory. The study suggested that reading a novel causes measurable changes in resting state brain connectivity, and these changes can last beyond the immediate reading experience. On reading days, connectivity involving these regions spiked. Why would reading activate sensory and motor areas? The researchers proposed embodied semantics. When you read about a character running, your motor cortex activates as if you're planning to run. When you read about someone touching velvet, your somatosensory cortex lights up as if you're feeling texture. The brain doesn't fully distinguish between reading about an action and preparing to perform that action. This phenomenon is called embodied cognition. Metaphors intensify it. A 2012 study at Emory by Lacey and colleagues showed that reading tactile metaphors like "she had a rough day" or "he has a velvet voice" activated primary somatosensory cortex. Reading literal sentences did not. The brain interprets metaphorical language by simulating the physical sensation. This places the reader inside the protagonist's body. It alters motor and somatosensory connectivity, not just during reading, but in resting state afterward. 246 data sets from 81 children. Accelerated longitudinal design. Some kids scanned once, some twice, some 3 times over several years. Mean age, 4.6 years. These kids couldn't read yet. They were pre-readers. Researchers measured pre-reading abilities using phonological processing tests and speeded naming tasks from the NEPSI-2 assessment battery, then diffusion MRI. They wanted to know, before reading instruction even begins, do children who will become good readers already have different white matter? Answer, yes. Children with stronger phonological awareness and faster naming speed had higher fractional anisotropy in specific white matter tracts, even before learning to read. The left arcuate fasciculus, the left superior longitudinal fasciculus, the inferior longitudinal fasciculus connecting occipital lobe to temporal lobe, the inferior frontooccipital fasciculus. These are the same tracts that mature during reading acquisition. But pre-readers who scored higher on pre-literacy tests already showed more organized white matter in these pathways. Does this mean reading ability is predetermined? Not exactly. The relationship is bidirectional. Genetics and early language exposure shape white matter development, which then supports reading acquisition, which then further strengthens white matter, creating a feedback loop. Published in the same December 2023 paper, Shared Reading Activities in Early Childhood Promote Phonological Awareness and Letter Recognition Skills. Children whose parents read to them from infancy had better phonological processing and more organized white matter by preschool age. Then structural MRI and diffusion tensor imaging. They specifically measured fractional anisotropy in white matter and cortical volume in gray matter regions associated with language. High-reading adults had significantly higher fractional anisotropy in the left superior longitudinal fasciculus and left arcuate fasciculus compared to low-reading adults. The differences persisted even after controlling for age, sex, and education level. But they also found cross-sectional correlations don't prove causation. Maybe people with naturally stronger white matter read more because it's easier for them. To test this, they brought back a subset of participants. Longitudinal follow-up. 60 adults scanned at baseline and again 6 months later. Between scans, half received no intervention. The other half enrolled in a 6-month intensive reading program. Average 4 hours per week, guided reading with comprehension exercises. After 6 months, the reading intervention group showed increased fractional anisotropy in language-related white matter tracts compared to the control group. The control group showed no change. This was the first evidence that increasing reading habit in adulthood can induce measurable white matter plasticity within months. But what about people who never learned to read as children and learn as adults? Extreme plasticity test. University of Sao Paulo, Brazil, and Collège de France, Paris, published June 2017 in Mind, Brain, and Education. Single case study. One 45-year-old Brazilian man, native Portuguese speaker, right-handed, no neurological disorders, completely illiterate, never attended school, couldn't recognize letters. He volunteered for an unprecedented study, 20 functional MRI scans over 23 months, about 1 scan per month. At baseline, he couldn't read at all. Researchers exposed him to words during the first scans. His brain showed no activation in reading-related regions. Zero activity in the visual word form area. That's a region in the left occipitotemporal cortex that expert readers use to recognize written words as a category distinct from faces or objects. But over 23 months, this man received intensive literacy training. He learned the alphabet. He learned phonics. By the end, he reached a modest level of alphabetical decoding. Not fluent, but functional. The MRI scans tracked his brain month by month. Gradually, activation began appearing in the left occipitotemporal cortex. The visual word form area emerged. Where there was nothing at scan 1, by scan 20, there was robust, consistent activation to written words. But here's the twist. As the visual word form area grew stronger, activation to faces in the same region decreased. Before literacy, the left ventral occipitotemporal cortex responded to faces. After literacy, the same tissue repurposed itself for reading, and face responses shifted slightly. This wasn't a perfect trade-off. The man didn't lose the ability to recognize faces. But the brain's limited cortical real estate forced specialization. Reading carved out territory, and faces adapted. Published in the same study, early stages of adult literacy learning recruited right hemisphere regions more heavily than left. By the end of training, activation became more left lateralized, similar to skilled readers. Frontal areas associated with working memory and executive control remained highly active throughout, suggesting adult literacy learning requires sustained effort and cognitive control even after basic decoding is achieved. 20 age-matched skilled readers as controls, published May 2020 in Neurological Research and Practice. Both groups received resting state functional MRI before and after a 1-year literacy training program. Only the illiterate group received training. The training program was called Alpha Plus. It focused on phonological awareness, letter-sound correspondence, word decoding, and reading fluency. Before training, functional illiterates showed increased intrinsic functional connectivity in a left frontoparietal network and in the basal ganglia network compared to skilled readers. Intrinsic functional connectivity means correlation between spontaneous brain activity in different regions when you're not doing a task. Higher connectivity in these networks suggested functional illiterates were recruiting more cognitive control and motor, planning resources even at rest, possibly reflecting compensatory strategies for their reading difficulties. After 1 year of training, the functional illiterates showed significant decreases in connectivity in these same networks. Their brains started looking more like skilled readers' brains. The frontoparietal network connectivity dropped, basal ganglia network connectivity normalized, reading test scores improved, the correlation was clear. As reading ability increased, the brain stopped overactivating compensatory networks. It became more efficient, published in the same study. Skilled readers showed no change in brain connectivity over the same 1 year period, confirming that the changes in functional illiterates were due to literacy training, not time or practice effects. Published August 2014 in Neuro Report. Longitudinal MRI study. 16 typically developing children aged 5 to 15. Mean age 10 years. Each child received 2 structural MRI scans. Mean interval between scans, 2.19 years. Almost 26 months. Between scans, children completed reading assessments. Word reading accuracy, reading fluency, rapid automatized naming. These tests measured different aspects of reading skill. The researchers measured cortical gray matter volume changes in 5 bilateral reading-related regions: inferior frontal gyrus, inferior parietal lobule, superior temporal gyrus, supramarginal gyrus, fusiform gyrus. These are the classic reading network regions identified in hundreds of functional MRI studies. Here's what they found. Children who performed better on word reading, fluency, and rapid naming at baseline showed greater volume decrease in left inferior parietal cortex and left inferior frontal cortex between time 1 and time 2. Volume decrease, not increase. Better readers had more cortical thinning. This matches the Belgian study from earlier. Cortical thinning in reading regions during childhood reflects synaptic pruning and myelination. The brain eliminates weak connections and strengthens important ones. The cortex gets thinner but more efficient. Published in the same neuro-report paper, these relationships were independent of age and total cortical gray matter volume change. That means it wasn't just general brain maturation, it was specific to reading-related regions and correlated with reading skill. Adolescent Brain Cognitive Development Study. 11,875 children aged 9 to 11 at enrollment, recruited between September 2016 and November 2018 from 21 sites across the United States. Longitudinal design. Follow kids through adolescence with annual or biannual MRI scans, cognitive tests, behavioral questionnaires. Published February 2025 in Scientific Reports. Researchers analyzed data from 8,125 participants who passed quality control for both MRI and cognitive measures. They asked, what are the associations between reading habits, television viewing habits, brain morphology, and cognitive performance? Parents reported how many hours per week their child reads books for pleasure and how many hours per week their child watches television. Researchers correlated these hours with cortical area measurements from structural MRI and scores on cognitive tests. Greater reading time associated with higher cognitive performance across multiple domains. Vocabulary, reading comprehension, working memory, processing speed. Greater television viewing associated with lower cognitive performance, but the effect was much smaller than reading's positive effect. Brain structure findings. Greater reading time associated with regionally selective increases in cortical area. Where? Lateral temporal cortex, inferior parietal cortex, inferior frontal cortex. These are the reading network regions again. Increased cortical area means more surface tissue in these regions. Greater television viewing associated with decreased cortical area in some regions, but the effect was weaker and less consistent. There was spatial overlap. Regions where reading increased cortical area and television decreased it partially overlapped in lateral temporal, inferior parietal, and inferior frontal lobes. But reading also showed unique positive associations in ventral temporal cortex, inferior temporal cortex, and cingulate cortex. Regions related to visual word recognition, semantic processing, and attention. Published in the same Scientific Reports paper, the correlation between reading hours and cortical area remained significant even after controlling for socioeconomic status, parental education, and screen time. This suggests reading has an independent structural effect on the brain. In gray matter, where cell bodies sit close together, water diffuses equally in all directions. Isotropic diffusion. In white matter, axons run in parallel bundles. Myelin sheaths wrap around axons, creating barriers. Water molecules can move freely along the axon, but not perpendicular to it. Anisotropic diffusion. Diffusion tensor imaging sends magnetic pulses through the brain and measures how water molecules move in different directions. From this, it calculates fractional anisotropy, a value between 0 and 1. 0 means water diffuses equally in all directions, isotropic, like in cerebrospinal fluid. 1 means water diffuses only in one direction, maximally anisotropic, like in a perfectly organized white matter tract. In human white matter, fractional anisotropy typically ranges from 0.2 to 0.8. Reading studies consistently show fractional anisotropy in the left arcuate fasciculus ranges from 0.45 to 0.65 in skilled readers. In poor readers or pre-readers, it ranges from 0.35 to 0.55, 10 to 15% lower. Published in a meta-analysis covering 1998 to 2024, this difference is one of the most replicated findings in reading neuroscience. Now, talk about what happens during reading itself, not just at rest. UC Davis, California. Published April 2016 in Journal of Neuroscience. Revolutionary method. Functional MRI combined with eye tracking. Lying in the scanner, participants read paragraphs of natural text on a screen. Eye tracking device registers which word they're looking at in real time, called fixation-related functional MRI, or FIRE fMRI. Before this, fMRI studies of reading used isolated words flashed one at a time, not natural reading. With FIRE fMRI, researchers see brain activation for each word as it's read in context, 2 to 4 words processed per second. Normal reading speed. They tested semantic grounding. When you read the word hammer, does your brain activate motor regions associated with using tools? The grounded cognition hypothesis says yes. The symbolic representation hypothesis says no, that word meanings are abstract symbols disconnected from sensory or motor experience. They measured brain activation to manipulable nouns versus non-manipulable nouns during natural paragraph reading. Manipulable nouns: hammer, scissors, brush. Non-manipulable nouns: mountain, idea, weather. Results: left anterior, inferior parietal lobule, and posterior inferior temporal gyrus showed significantly higher activation for manipulable nouns. These regions are associated with action performance and action semantics. Previous studies with isolated words found the same pattern, but this study proved it happens during rapid, natural reading of connected text. Even when reading a paragraph at normal speed, the brain briefly simulates the motor actions associated with manipulable objects. Published in the same Journal of Neuroscience paper, the effect occurred for every manipulable noun, not just when the word was relevant to the sentence meaning. Even if the paragraph wasn't about tools, reading the word hammer activated tool use regions, automatic, unavoidable. Semantic map of the entire cortex, not just reading regions, the whole brain. 9 participants. Each spent hours inside a functional MRI scanner. They listened to stories from the Moth Radio Hour podcast and separately read the same stories. Stories were time-coded. Every word matched to a specific time point. Brain activity measured throughout. Researchers used statistical modeling to map which brain areas responded to which semantic categories. Visual words like red and bright activated visual cortex. Touch words like soft and rough activated somatosensory cortex. Emotional words like happy and angry activated medial prefrontal regions. Social words like friend and talk activated areas involved in theory of mind. Numeric words activated parietal regions. Locational words activated regions involved in spatial processing. Violent words activated regions associated with action and threat detection. They created a 3-dimensional interactive brain map, color-coded. Thousands of words plotted across the cortex based on where they activated. The maps from listening and reading conditions were virtually identical. Reading activated the same semantic map as listening. This means reading isn't just visual processing followed by language processing. Reading accesses the full distributed network of conceptual knowledge across the entire cortex. The visual word form area recognizes the letters. Phonological regions decode the sounds. But semantic access spreads everywhere, activating every region relevant to the word's meaning. Published in the same Nature Neuroscience paper, at least 1/3 of the cerebral cortex participated in semantic processing during reading, not just language areas. 1/3 of the brain. That's why reading feels immersive. It recruits most of the cortex. Published November 2023 in Communications Biology. Adults with dyslexia and age-matched controls performed reading tasks during functional MRI. Researchers measured both brain activation and functional connectivity. Dyslexic adults showed hypoactivation in left hemisphere reading regions, left temporal parietal cortex, left occipitotemporal cortex. These regions showed reduced activity compared to controls, but they also showed disrupted functional coupling. The connections between left temporal parietal and left occipitotemporal regions were weaker. These regions weren't communicating efficiently during reading. Functional connectivity analysis revealed dyslexic readers relied more on a dorsal reading route. Dorsal means through the top of the brain, involving parietal regions and phonological decoding. Skilled readers use both a dorsal route for decoding unfamiliar words and a ventral route for rapid whole word recognition. Ventral means through the bottom, involving temporal and occipitotemporal regions. Dyslexic readers showed weaker ventral root connectivity and stronger dorsal root activation, suggesting they're stuck decoding every word phonologically instead of recognizing words automatically. The study also found increased involvement of right cerebellum in dyslexic readers. The cerebellum supports motor coordination and procedural learning. Its involvement in dyslexia remains debated, but the connectivity data showed dyslexic readers had stronger reading-related interactions with right cerebellar regions than controls. Compensation mechanism. The cerebellum might be supporting extra rehearsal or procedural aspects of reading when the typical left hemisphere network isn't functioning efficiently. Family risk means a parent or sibling has dyslexia, genetic predisposition. Researchers scanned these at-risk children before they received any reading instruction and compared them to control children with no family history. Then they followed the children longitudinally and measured reading outcomes at age 7 or 8. Question: do pre-readers, who will later develop dyslexia, already show different brain structure? Answer: yes. The most consistent finding: differences in left temporal parietal regions. Children who later developed dyslexia had reduced gray matter volume, or altered white matter organization, in left temporal parietal cortex even before learning to read. These regions are associated with phonological processing, mapping sounds to letters. The phonological deficit theory of dyslexia proposes that dyslexic individuals have impaired phonological representations. These MRI findings support that theory. The neural substrates for phonological processing are atypical before reading instruction begins. Published in the same meta-analysis, differences in left ventral occipito-temporal regions, the visual word form area, also appeared in some pre-reading at-risk children, though less consistently than temporoparietal differences, some evidence for early deficits in auditory perceptual regions. The take-home? Dyslexia has neural signatures detectable before symptoms appear. This opens the door for early screening and intervention during the period of maximal brain plasticity. White matter was considered passive infrastructure, just cables. But recent studies using high-field MRI scanners, 7 Tesla or higher, found blood oxygen level dependent signals in white matter. White matter has its own functional activity. Published November 2025 in Communications Biology, researchers developed the gray-white matter heterogeneous fusion network, a method to measure functional connectivity between gray matter regions mediated by white matter bundles. They used resting state fMRI data from 6 independent datasets: Human Connectome Project, CAMCAN Aging Study, others. They found that white matter doesn't just structurally connect gray matter, it actively participates in functional communication. White matter bold signals show task-related and resting state fluctuations. Connectivity between 2 gray matter regions is strengthened when they share connections through the same white matter bundle. The paper introduced a new metric, covariance of gray matter-white matter interaction profiles. Basically, if 2 brain regions both interact strongly with the same white matter tract, they're functionally connected through that tract. This framework revealed that 89% of the brain's white matter is shared between multiple resting state networks. 16% of white matter is shared by at least 7 networks. White matter isn't a set of private cables. It's a shared highway system where many networks overlap. Lesions in these high overlap regions would impact multiple cognitive functions simultaneously. Published in the same Communications Biology paper, the method showed fair test-retest reliability over short intervals and slight to fair reliability over long intervals. It's sensitive to individual differences, aging effects, and autism spectrum disorder symptoms. White matter functional connectivity is a legitimate, measurable phenomenon that adds another layer to understanding brain networks. Published July 2023 in Nature Communications Biology. Researchers created an atlas of 30 resting state networks using Human Connectome Project data. They jointly mapped the white matter and gray matter contributing to each network. Previous studies identified gray matter nodes of the reading network, inferior frontal gyrus, superior temporal gyrus, inferior parietal lobule, fusiform gyrus. This study added the white matter. Arcuate fasciculus connects inferior frontal and superior temporal regions. Superior longitudinal fasciculus connects frontal and parietal regions. Inferior longitudinal fasciculus connects occipital and temporal regions. Inferior fronto-occipital fasciculus connects frontal and occipital regions through ventral pathways. The atlas showed these white matter tracts aren't dedicated to reading. They're shared by language network, attention network, visual network, and memory network. Reading relies on coordination across multiple networks, not a single isolated circuit. The overlap explains why reading disorders often co-occur with attention deficits or memory problems. The networks share infrastructure. Published in the same Nature Communications Biology study, most white matter lesions, from stroke or multiple sclerosis, impact multiple networks simultaneously because of shared white matter. A lesion in the left superior longitudinal fasciculus might impair reading, spatial attention, and working memory all at once. Structural MRI and diffusion tensor imaging at baseline. Reading assessments, subset of 90 participants, scanned again 1 year later. Cross-sectional analysis showed fractional anisotropy in left superior longitudinal fasciculus and left arcuate fasciculus positively correlated with reading comprehension scores across the entire age range, 8-year-olds to 39-year-olds. The correlation was strongest during late childhood and adolescence, ages 8 to 18, when reading skills are still developing rapidly. But it persisted into adulthood. Adults with higher reading comprehension had higher fractional anisotropy in these tracks. Longitudinal analysis showed fractional anisotropy in reading-related white matter increased over 1 year in children and adolescents who reported frequent reading, decreased slightly in those who reported infrequent reading, and remained stable in adults. And this suggests white matter plasticity related to reading continues through adolescence and stabilizes in adulthood unless reading habits change dramatically. Published in the same paper, the relationship between white matter and reading is dynamic, not static. Oligodendrocytes, the cells that produce myelin, actively monitor neural activity and adjust myelination. In response, when a white matter tract is used frequently, oligodendrocytes increase myelin thickness, speeding signal transmission. When a tract is underused, myelination decreases. Reading provides consistent, repeated activation of language-related white matter tracts, triggering activity-dependent myelination. No large-scale MRI studies tracked these individuals specifically, but we have proxies, professional literary scholars, lifelong avid readers, published April 2025 in Neuroscience and Biobehavioral Reviews, meta-analysis of 163 fMRI studies on reading in adults, coordinate-based meta-analysis, every study reported which brain regions activated during reading tasks. Researchers compiled all the coordinates and looked for convergence. The left inferior frontal gyrus showed consistent activation across word reading, pseudo-word reading, and sentence reading. Subregions within inferior frontal gyrus showed differential engagement. Pars opercularis, the posterior part, activated for phonological decoding. Pars triangularis, the middle part, activated for semantic retrieval. Pars orbitalis, the interior part, activated for sentence comprehension and integration. The left posterior temporal cortex showed consistent activation for all reading levels. Left occipitotemporal cortex, the visual word form area, activated for letter and word recognition. The cerebellum, specifically left cerebellar lobule 6 and right crus 1, showed consistent activation during reading. The cerebellum supports motor sequencing, prediction, and error correction. During reading, it might coordinate eye movements, subvocal articulation, and predictive processing of upcoming words. Right hemisphere contributions appear primarily for pseudo-word reading, which requires effortful decoding, and for lexical decision tasks that require explicit judgments. Silent reading, the kind you do when reading a book, showed more consistent left hemisphere dominance, published in the same meta-analysis. Overt reading, where you read aloud, activated auditory cortex and motor cortex more than covert reading. Covert reading relied more on multiple demand regions in frontal and parietal cortex, possibly reflecting internal monitoring and comprehension processes without motor output. But there's something the studies don't capture well. Real-world reading, novels, textbooks, news articles. Most fMRI studies use isolated words or short sentences. Artificial. Published July 2020 in Scientific Reports, researchers tried naturalistic reading in the scanner. Participants read scientific texts paragraph by paragraph while eye-tracking recorded fixations, fixation-related fMRI. Each fixation triggers a specific brain response. The study manipulated the knowledge structure of the texts. Some texts had optimal knowledge structure with clear hierarchical organization. Other texts had suboptimal knowledge structure with tangled connections between concepts. They also measured individual differences, reading ability, electronic device usage habits. Participants with higher reading ability and lower screen time showed greater activation in left frontal pole cortex and supplementary motor area when reading suboptimal texts. These regions support multitasking, cognitive control, and integration of complex information. Essentially, better readers recruited more neural resources to handle poorly structured text. Participants with higher electronic device usage showed less activation in key integrative regions, particularly left inferior parietal lobule. Heavy screen users had reduced engagement of brain areas necessary for deep comprehension. Published in the same Scientific Reports paper, habitual electronic device usage might adversely affect high-level cognitive processing required for scientific text comprehension. The data suggest frequent switching between apps, rapid scrolling, and fragmented digital reading disrupt the neural networks that support sustained integrative reading. Reading comprehension associated with more left lateralized activation and left inferior occipital cortex activation, including fusiform gyrus. That's the visual word form area. Reading requires visual processing. Listening comprehension associated with extensive bilateral temporal cortex activation and more overall activation across the whole cortex. Listening recruits both hemispheres more equally. The auditory input is processed bilaterally. But despite these modality differences, a common core of modal regions activated for both reading and listening. Left inferior frontal gyrus, left middle temporal gyrus. These regions process meaning regardless of whether it comes from text or speech. Published in the same study, readers with lower working memory capacity showed more spillover to right hemisphere regions and more prefrontal activation, reflecting compensatory executive control. Readers with higher working memory capacity showed more activation in left angular gyrus and precentral gyrus, possibly reflecting efficient phonological rehearsal. Individual differences matter. Reading recruits different networks depending on cognitive capacity. Reading changes brain structure and function in measurable ways. White matter tracts become more organized. Gray matter regions specialize for reading thicken during childhood, then refine and thin during adolescence, reflecting synaptic pruning and myelination. Functional connectivity between language regions, visual regions, memory regions, and even motor and sensory regions strengthens. These changes don't require decades. The Emory study showed changes within days. The Tohoku study showed changes within 6 months. The Brazilian literacy study showed a complete reading network emerging in an illiterate adult within 2 years. The brain is plastic. It will reorganize based on what you do repeatedly. If you read frequently, you're training networks that support not just reading, but comprehension, perspective taking, semantic knowledge, and embodied simulation. If you don't read, those networks remain underdeveloped or atrophy from disuse. Published in multiple papers, screen time, especially passive video watching or rapid scrolling, is not a substitute. MRI studies show screen time without caregiver interaction associates with disorganized white matter in preschool children. Electronic device usage in adults correlates with reduced activation in integrative processing regions during reading tasks. Screens aren't inherently bad, but the way most people use them, fragmented, distracted, shallow, does not engage the brain's reading network deeply. Published in the ABCD study and multiple other longitudinal studies, reading habit during childhood predicts cognitive performance and brain structure in adolescence. Schools that prioritize sustained, deep reading, not just decoding drills, but actual books, actual stories, actual sustained attention build stronger neural architecture in their students. Interventions work. The functional illiterates in Germany who underwent literacy training showed normalized brain connectivity after 1 year. Dyslexic children who receive intensive phonological training show increased activation in left hemisphere reading regions and improved reading scores. Brain plasticity means remediation is possible at any age, though earlier is better because plasticity decreases with age. You're not done developing. White matter plasticity continues into your 20s, and functional connectivity remains adaptable throughout life. If you read regularly, you maintain and strengthen reading networks. If you stop reading for years, those networks weaken. Published in aging studies, older adults who continue reading show less cognitive decline. Reading provides cognitive reserve, a buffer against age-related neural degeneration. The semantic knowledge, vocabulary, and comprehension networks built through a lifetime of reading remain active and support other cognitive functions like memory and reasoning. Reading is cognitive exercise. Published in multiple neuroplasticity reviews from 2024 and 2025. Neuroplasticity, the brain's ability to adapt structurally and functionally, persists throughout adulthood. It's not just for children. Adults can induce structural changes through sustained behavioral changes. Reading is one of the most accessible and powerful tools for driving beneficial brain changes. No expensive equipment, no prescriptions, just books and time. Few studies tracked lifelong reading habits across decades. Researchers at multiple institutions are now launching multi-decade longitudinal studies to follow individuals from childhood to old age, measuring brain structure, reading habits, and cognitive outcomes every few years. Early data from these cohorts hint that cumulative reading exposure across a lifetime predicts cognitive reserve, dementia risk, and quality of life in older adulthood more strongly than any single cross-sectional measure. But the full data won't be available for another 20 or 30 years. The participants are still being tracked. What we do know, every MRI study that measured reading and brain structure found associations. Higher reading frequency, more organized white matter, more reading skill, larger gray matter in reading regions, more lifetime reading, better preserved cognitive function in aging. The effect sizes vary, the specifics differ, but the direction is always the same. Reading is not neutral, it's not passive. It's a brain-building activity with permanent structural consequences. And inside your skull, networks fire that wouldn't otherwise. White matter organizes, gray matter specializes, connections strengthen. The changes persist for hours, days, maybe longer. Do it again tomorrow, and the day after. The changes accumulate. 6 months from now, your brain will have different white matter structure than it does today if you read frequently. MRI scanners caught this. Scientists mapped it. The data is final. Frequent readers don't just know more words. Their brains are physically, measurably, structurally different.